Good day. My name is Rob Ferl, and it's my distinct pleasure today to give you some insights into the topic of space molecular biology, the biology of living in and adapting to space flight. It's a topic that is really fundamentally interesting to me, and it allows us to understand and to really appreciate what it takes to live in space, what it takes as a biology, as an organism, as a person to adapt to a very strange environment, but also as a biologist, it allows us to test some questions and some notions about the limits of biological adaptivity. It's one thing for us to talk about what it takes to live on a place in the surface of the earth where organisms on earth have had a chance to explore those environments and find out how difficult it is to adapt to that environment. But it's a whole nother thing to ask a question about what does it take to adapt to an environment that has never been within the evolutionary background of an organism. So when we take organisms to space, when in our case we take plants to space, we are really asking some fundamental questions about the limits of adaptability. How many kinds of tools are in the tool chest of an organism that can be brought to bear on a totally strange and a totally novel environmental challenge? So in space biology, we really want to ask the question, what will it take to allow our biology to live on another planet, to live off the surface of the Earth, to live within an exploration vehicle like the International Space Station or the transit vehicle that humans will take to Mars? And by learning that, we understand the limits of life on Earth, and we begin to understand our bigger role in the universe as a whole. So as molecular biologists, we study the adaptation processes as plants move to this new environment, that of living in space. We hope that informs us on how we can help support astronauts in the future as they move further and further from the surface of the Earth, but it also helps us understand our place in the universe by helping us define the limits of how far terrestrial biology can adapt to novel environments. And in particular, as plant space biologists, we are allowed a very, very special place in the exploration panorama because plants, as indicated by this montage of the movie The Martian and a plant put there in support of The Martian, you can see that plants are part of the exploration sort of think because plants, just like on the surface of the Earth, in a space vehicle or on Mars, plants would supply oxygen, they'd supply carbohydrates and recycle water, while also taking in the CO2 and other wastes of the human in order to do life support. And as such, we in plant molecular biology can both support the exploration agenda while we ask those fundamental biological questions about what it takes to move a terrestrial organism into a totally novel environment. So as plant molecular biologists, we both support the human spaceflight agenda by providing food and clean water based on energy inputs such as light. And we also feed the basic understanding of what it takes to adapt to a totally novel environment. For the time being, all of our exploration vehicles will be in a weightless environment. Now, this is a fanciful picture of what a space greenhouse might look like, but there are a couple of key features that are important. First off, there's no artificial gravity. There is nothing in the plans of any current international spaceflight effort to produce artificial gravity while on orbit. So plants and people and everything associated with the environment are going to be in a place where there is no gravity. Now remember, gravity is one of those big fundamental forces that have shaped every organism on the surface of the earth with regard to its structure and its function, especially plants who are dedicated to growing their leafy parts away from gravity. I think it's fundamentally interesting to ask the question of what happens to plants and how do they grow in the absence of that force. So on the International Space Station, we've been able to do quite a few actually pretty interesting experiments that address this question. In this picture, you can see astronauts from 2015 who are in the process of eating some lettuce that was grown on the space station. It was grown in this facility that is producing the bright light over here, and we're going to see a different view of this facility in just a minute. But basically, they were able to grow enough lettuce there to enjoy a fresh salad in space for the first time in an awfully long time. And what that does is it allows us to basically pull together some questions that really probe the nature of the adaptive process of living without gravity. First and most important thing to understand is that plants do grow largely normal in space. The leaves still grow towards the light. Roots still grow away from the light. And the cell structures and functions are basically the same. Photosynthesis is basically the same. I find that incredibly interesting that a plant, which has been so totally dedicated to growing in the presence of gravity throughout its evolutionary history, looks basically perfectly normal throughout the entire development process in space. Now, our experiments with molecular biology in space are based on small Arabidopsis plants that grow on these Petri plates. One of the cool things about this as an experimental approach is that the plants sitting on these Petri plates don't need a complex environment or support system to live. They don't need nutrient delivery. That's taken care of by the auger. They don't need to manage water. That's also taken care of by the auger. And when wrapped in black cloth here, the dormant seeds can be sent into space such that every developmental process from the germination of the seed on through to complex development such as the production of leaves and flowers can take place in space. So the plants we are going to be comparing from being in space to being on the ground are going to be plants that enjoyed their entire lifetime on the space station. Now off to the right here you'll see a specialized tube. This is basically the microfuge tube of the space station. It's a little bit bigger, and what it does is it allows us to put preservatives such as RNA later into space and to preserve plants taken off from these plates and put into these tubes in the space station such that experiments can then be brought back to Earth for gene expression analysis. Now remember that bright light with the astronauts on the space station when they were eating the lettuce? This is a different view of that bright light. This is the growth chamber that our plants are going to be sitting in, and these are the Petri plates that have been taken to orbit and put there. This is astronaut Peggy Whitson, and what she's doing is she's showing you the Petri plates that we have sent to orbit, and you can see she's unwrapping them. She's going to take a look at it, and here's your first indication right there that she's actually in space, because that Petri plate is just floating there. It's got nowhere to go. It doesn't move anywhere. It doesn't fall to the ground. And what you're going to see is that she's going to take some pictures of it. She's going to move it around a little bit, and then she's going to put it into the racks inside that growth chamber. Now, when she rotates around and gets in the way of the light, you'll see that the automatic camera that's taking the video will adjust the light, and you'll get a more realistic view of the chamber and the lighting in the space station at that time. That's coming right up here. You can see, there you go. Now it looks like a normal environment when the automatic lighting control isn't actively trying to sort of cut the iris down there. So that's basically the experimental system. Petri plates with seeds go up to space, and then they grow in space, they're harvested in space, and what we're going to do is take a look at some molecular biology of the root tip here to ask those fundamental questions about gene expression in space and allow us to address questions about that whole phenomenon of adapting to living in space. Among the various things that we can do is look at reporter gene activity. Here's an astronaut that is putting one of our plates into the microscope that's on the space station. From down here on Earth, we can control the microscope and take pictures, and what we do is do a series of gene expression studies using fluorescent tags. Here, for example, is a gene involved in auxin production in the root tips there. And you can see we can get fantastic views of the gene expression characteristics and cell morphologies in space compared to the ground. And in this particular instance, you can see while the cells are a little bit bigger over here and smaller down on the ground, the gene expression patterns for that particular gene are exactly the same in space as on the ground. So basically, we can understand that for that gene in that environment having to do with the production of an extremely important plant hormone, auxin, is basically the same on the Earth as it is in space. Now, the main thing that I want to leave you with is a discussion about the adaptive processes in space as revealed by gene expression studies. And again, I wanted to give you an impression of what it's like to do those experiments in space. Here's astronaut Jeff Williams. He's taking one of our Petri plates there. He's going to take a picture of it, and you can see that he's got the tube for depositing the plants. That tube is filled with RNA later, and what he will do after he takes a picture is he'll take the plants off the plate, put it in that tube, and in the next step, what he'll do is put the RNA later in contact with the plants so that they're preserved. He'll freeze them on orbit in the -80 freezer there, and then they'll come back to Earth so that we can do RNA-seq analysis on the genes that are expressed in any given tissue. And here's some of the take-home lessons from that. The first is that depending upon whether you're a leaf, a hypocotyl, or a root, you express different genes differentially to adapt to spaceflight. Here are the genes that are upregulated, here's the genes that are downregulated, and you can see that leaves, hypocotyls, and roots do not share a whole lot of genes in common when they adapt to spaceflight. Rather, each organ is finding its own adaptive path. And that's going to be an important thing to remember, that each organ has its own gene expression pathway to adapting to living in space. Moreover, and I look at the total numbers here, there are not a lot of genes involved in adapting to spaceflight. In fact, there's only a couple of hundred, and most of those hundreds have to do with mediating signals that normally are associated with gravity sensing and converting those into changes that are associated in for many of these plant genes that are associated with the cell wall and remodeling the cell wall so that it no longer has to be as thick or as heavy as it is in space as it is on the ground. So each organ has a different response to adapting to space. All of those responses seem somehow related to changing cell wall morphologies. And the important thing that I really want to leave you with as far as the adaptive process is to ask the question, in the root tip in particular, where gravity is sensed by plants here on the Earth, what are the changes in gene expression that happen there? And how are those changes in gene expression altered depending upon the genotype of the root? Here's Columbia, one of the favorite genotypes of Arabidopsis flown to space, and you can see it takes 297 changes in gene expression in the root tip to adapt to space. If we take a Phi-D mutant, a phytochrome D mutant, doesn't really matter what that is, but that's a light sensing mutant, a single gene mutation in Columbia, and fly that to space, it only takes 120 genes differentially expressed to adapt to space. If we send a different genotype, a different ecotype called WS, that genotype, when it goes to space, only has to differentially express 62 genes. This is a big difference. This is almost no effort at all. This seems like a big effort. And yet all three of these genotypes adapt to spaceflight just fine. So not only like the leaves and the roots, where the organ specificity determined a different path to the response to spaceflight, so too different genotypes take different paths to adapt to spaceflight. Spaceflight also is not a stress like cold or heat with a common set of very recognizable genes. Rather, each of the genes that is differentially expressed are related only in the vaguest ways associated with cell wall metabolism and other things associated with growth and development. There is no single set, very identifiable set of genes that characterize this spaceflight adaptive process. And we see the same information, the same level of information when we look at proteomics or methylation of the genome, of the epigenome in response to space. There is very little in common, very little that characterizes a single adaptive response to space. Rather, it is an organ-specific and genotype-specific adaptation to space. If we take those same gene expression profiles and overlap them, so that you can see that the number of genes that are in common between Phi-D, Columbia, and WS, you can see that those 18 genes that are in common are actually very few of the genes that are separate, that are differentially expressed overall. However, if we take a look at the genes that are absolutely 100% identifiable as associated with cell wall metabolism, you can see that each of these genotypes does a cell wall remodeling of one kind or another. Now again, that's genotype specific, but each of these black dots represents a gene associated with cell wall metabolism, at least as well as we can initially detect. Moreover, all of the genes that are associated with every one of these responses has something to do with signals involving shape or signals involving pathogen responses. Now, the annotations of plants, and especially plant genes, are well characterized for pathogen responses because those are easily studied events. But do remember, many pathogen responses have to do with disruptions in the cell wall. So we think that many of the responses that we are looking at that have the term pathogen response are actually cell wall remodeling responses. And the genes that are in common there, even if you look at those, they basically have some of the same characteristics. These genes that are upregulated in common among all those genotypes have to do with peroxidases, osmotins, and other things that are very likely, although not obviously, associated with cell wall metabolism. And all the genes that are upregulated together have to do with the interpretation of light signals in and around the genome of plants. So we suspect that what we're seeing here is the result of the adaptation response is a change in cell wall metabolism, mostly by downregulation of certain key genes. And we're seeing the upregulation of genes associated with light signaling. And from our perspective, that's likely because the plant is pulling heavily on light perception as a way to guide the directionality of its development, as opposed to using gravity. So the journey to spaceflight allows us to reinterpret the names of some genes and their annotations, and it's basically allowed us to understand that moving off the surface of the Earth is not a single response that is going to be very well recognized among all plants or all plant organisms, and there's certainly not going to be a recognizable gravity-related response, spaceflight-related response between animals and plants. But there are major lessons that we're learning about growing plants in space having to do with their metabolism, having to do with the limits of their adaptive capabilities. And we're probing using genetics and molecular biology the ideas that spaceflight responses can be modified, limited, or better understood so that we can even better support the exploration of humans into space by growing better plants on the surface of Mars or better plants in space to supply astronauts with food, while at the same time satisfying the curiosity of a pure molecular biologist who wants to know what happens when an organism goes into a totally novel environment and has to create an adaptive response to live there. Now, I have to say, as molecular biologists, we don't always get a chance to do all of the things we would really love to do. It would have been a real joy to be at this meeting with you and talking about this in the context of the personal relationships that often happen at meetings, but as sometimes happens for us, even as laboratory molecular biologists, we get to do some really interesting things. Here's my colleague Annalisa Paul and me in Antarctica working on the Eden ISS project. I encourage you to take a look at that. It's eden-iss.net is the online place to go look for this project, and we'll be with our European and other North American colleagues working on a plant growth system for feeding people in Antarctica as an analog for feeding people on the moon or Mars. Take care and best regards.